It's like a second coffee
All right,
cup.
hopefully
Yeah.
all right, so, as you guys all know, it'll be fun, it'll be exciting. Chance that you're excited for this test, chance to show all the things that you know, right? Uh-huh, okay.
Hey, we do get a note card.
Wait.
You have a note card, that's right. One thing to remember is that you do get a note card. 3x5. 3x5 is yay big, not real big.
3x5.
Right?
Yeah.
Sure. I don't know what a regular flash card looks like.
Just the normal.
Just a normal note card. Yeah, like that. Perfect.
They're in a circle.
Like that.
3x5 meters.
Yeah, so one note card, both sides if you would like both sides. I mean, at this point, you know, you should be able to kind of figure out what you don't know and what you know. And if you have problems with that, if you have problems with anything, I know that there's actually there's a good number of people that did not hand in the translation assignment or the last lab, which means that I think there's some people having trouble with this. If you are one of those people that are having trouble with this stuff, it would be nice if you talk to me before the test on Thursday. And my Thursday mornings are pretty darn busy. So that leaves you with today, Wednesday. Right? Okay. Well, after this class, I've got another class. I'm free until 11 after that. Got a meeting at 11. I've got you guys at 12. I'm free until 3 after that. Okay? Those are the times that I have. Thursday, I have a lab going on in the early morning. And you might be able to catch me, but I would prefer that if you do it, you should do it today. Otherwise, you're stuck with what you know and what you don't. There are tutors available as well. So you have to get in very quick. Okay, this is test, that's our test number 2.
Yeah.
I mean, realize there's enough points in this class that you can flub up one test. But if you're working on flubbing up a second test, you really don't want to do that. I would not want to do that. I don't know what you can do whatever you want. So just try not to flub it up. I prefer it if everybody's above the C before I put in grades. I think they can do it. Also, remember, there's a plate of ballot brownies on the line. If you guys all get the labels of 5' and 3' labeled correctly on all of your DNAs and RNAs, okay? And N terminus to C terminus on all of your proteins. Got it? Okay. I have never had to pay up. I would be thrilled if I have to make brownies.
Can we do like lemon squares? If we get-
No,
We don't want brownies. One thing to announce, on Tuesday, we have a very special guest. They're gonna be in this room with the pediatric model of a child pediatric, you know that, who's undergoing asthma. So a mannequin that's undergoing asthma. You're gonna learn how to treat an asthma. There are slots open from 2 to 3, 3 to 4, and 4 to 5. I believe that if you have Dr. Ryder's class on Tuesday afternoon, Dr. Ryder's lab on Tuesday afternoon, who's got that lab? Is that A&P or is that anatomy, physiology? That's just physiology. Yeah, so if you're in physiology, you're already enrolled. If you're in hematology, you're already going because we're going at 4. If not, we would love it if you would sign up. There are still open slots. If you do not know how to access the open slots came in an email from Kelly Schoch. Did not say her name correctly. And she has sent out another email, I believe. Because the first one came over break and hardly anybody signed up. So if you guys would sign up, that would be great. I'd hate to spend a lot of money on these guys and then have nobody come. They're cool. It's cool. It's the simulation truck people. I know what they're talking about. This should be right up your alleyway if you are a biology major of any sort. Even if you're not in healthcare. Okay. It's sponsored by Tri Beta. With a little help with the Health Professions Club. Tri Beta. And if you want to join Tri Beta, I'm going to send out those forms later in the week. So if you're interested in joining Tri Beta, please fill out a form and get it back into me. Okay. All right, so now halfway through the class, let's get on to our show. All right, last time we were talking about combinatorial control, which is basically the idea that at the promoter of eukaryotes, there are lots of things that are involved in controlling this promoter. So there are multiple sites upstream. Remember upstream? Okay, this is +1 where your transcription starts. Okay, this right here is called what? Okay, and what binds to the title box? CBP, okay? Okay, and then over here, there's a bunch of things called activators or silencers that will bind to control the expression of genes. Multiple ones, and as I said before, this does not go up and down like flipping on a switch. It goes up and down like a dimmer switch. So if I've got an activator here, that's probably just enough to turn it on a little bit. But if I add several more activators and kick off a silencer, then it goes up a lot. Okay. So that's going to be controlled by gene expression, those gene expressions are going to be controlled by signal transduction pathways in the cell. So signal transduction pathways you've learned about before. And they're basically ways for the cells to respond to things coming into the cell from the outside or to things inside the cytoplasm.
I have to install Office. Thanks a lot.
We're not going to go through signal transduction pathway. That's kind of beyond the scope of this class. But remember what happens is you basically have some signal coming in from the outside. Okay, that binds to a receptor. Turns on some kind of transduction pathway. At the end of it, you basically, what you will do is you will activate a transcription pathway. One of these guys, an activator or a repressor, silencer or repressor. Those things basically will then cause the cells to either turn on a gene or turn off a gene. Or slightly dim a gene or slightly activate a gene more. Okay, you can have a signal coming from outside. You can also have signals coming from the inside, binding to internal receptors. But either way, they send a signal to the nucleus to turn on transcription factors. Okay. Sometimes those transcription factors are stuck in the cytoplasm until they get a response that allows them to go to the nucleus. Sometimes they get phosphorylated. That allows them to go to that allows them to start transcription. Or they get modified in some other way. Okay. So here is an example. This is not an example that you need to know, particularly not for this test, but even for the next test. Not an example I'm going to ask you questions about, but this is called the NF-kappa B pathway. So NF-kappa B is the transcription factor right here. Okay. Normally, it's held in the cytoplasm by a factor that's called I-kappa B, which is the inhibitor of NF-kappa B. So NF-kappa B, kappa is a small k that you find in Greek letters. Nuclear factor kappa B, basically is what it is.
Get a bunch
Okay.
of
Held in the cytoplasm by I-kappa B. Okay. But when you get a signal coming down the cell, what happens is I-kappa B gets phosphorylated by a kinase. So IKK is the kinase right there. Phosphorylates NF-kappa or phosphorylates I-kappa B. I-kappa B then basically becomes a fluid. So I-kappa B gets basically degraded. NF-kappa B goes to the nucleus and it turns on gene expression. And these are genes that are going to be involved in growth or in the immune system response. But that allows you to basically respond to TNF up there, which is basically a cytokine. Comes down here, it allows you to basically respond to that by turning on NF-kappa B. This is one of the pathways that your cells use to turn on an inflammatory response. Okay? So that's basically what we're doing. So basically what's happening here is in your cell, you've got competition from all of these different receptors that are binding to different factors. So maybe this one's binding and that one's binding. This is a different signal transduction pathway that turns on a different transcription factor. Okay? All of those work together to control the gene. So if you've got multiple transcription factors turning on activators, the gene goes up really a lot and you get a lot of transcription. Got a signal transduction pathway that's activated. It turns on a repressor. That will silence the gene. So maybe this repressor here. This repressor here might overlap with this activator site. And maybe it kicks off the activator. And maybe that turns off your gene. So it's very complex, like I told you before. This is not like red light means stop, green light means go. This is like green light means go, but you shouldn't run over the pedestrians with the crosswalk. Right? Like, does that make sense? Okay, so this is basically your cell basically trying to integrate multiple signals on one promoter to then try to turn on one gene. That goes on in thousands of promoters at once inside of your cells. Some promoters are always off, some of them are always on. A lot of them are regulated by whatever signals are coming in. That make sense? Okay. That's a really shallow explanation, even though it sounds deep, it's a really shallow explanation of everything going on in the cell. It's kind of cool though, isn't it? Because your cells are very complex, but they're responding to these modular signals that are basically either you bind to a receptor or you don't bind to a receptor. Okay, so if I use this, I can basically say how much energy is in the cell. Are there growth factors being produced by other cells? You know, you can basically tell what's going on in the cell by using all of these receptors to basically tell you what molecules are being produced. All right, so now basically we've done the promoters. I tell you the promoter is very complicated. And we have all this stuff going on. But now we turn on this promoter and RNA polymerase is coming down, striking down the track, making a RNA. Is my RNA molecule. Okay, this is the tail, phosphorylated tail of RNA polymerase. And here's my RNA. Okay. The RNA itself can be regulated. So it can be regulated by alternative splicing, which we talked about a little bit before. It can be edited, which we really won't talk about much. It can be either stable or unstable. It can have interference. Or we can even start talking about translation regulation. Okay. Some of this is stuff that we just recently discovered within the last 20 years or so. So a lot of this is kind of new stuff. The first one basically is this idea of post-transcriptional regulation by alternative splicing. And this is really important because most of your genes inside your cells are potentially alternatively spliced. In some instances, they will keep a particular exon. And in some instances, sometimes, let me just say that better, keep a particular exon, sometimes they'll get rid of it. So here's two versions of the same gene. This one has 1, 2, 4, 5, 6, 8, 9, 10, and 14. You see it skipped basically 11, 12, and 13, okay? And this one basically goes 1, 3, 4, 5, 6, 8, 9, 10, 11, 12, okay? The red ones here are always found, they're constitutive. They're always found in the RNA. They're always translated into protein. And the green ones are sometimes in the RNA and sometimes not. It will depend upon the conditions of the cell and it will depend upon the cell type. Almost all of the genes that in your body can be alternatively spliced at some point or another. Okay, that gives us, even though we only have about 20 to 30,000 genes, that gives us a multitude of different patterns that we can produce. So we have a multitude of different proteins that we can produce because they can be alternatively spliced. So you can see that this particular gene, this is tropomyosin, in smooth muscle cells it has one pattern. It's striated muscle cells that has a different type. Striated muscle is a little bit different. How is striated muscle different than smooth muscle? Make Dr. Ryder proud. or Dr. Rust. I'm sure he taught you about muscles at some point. What would you find smooth muscle? Organs, yeah. Okay. Where would you find striated muscles? Muscles, okay? What's the difference?
Stronger. Smooth is involuntary.
Sorry?
Control.
Voluntary, yeah, whether you control it or not, whether it can fatigue or not, right? So there's also a few differences between them. So I would assume that these different exons basically allow for more conscious control in the terms of striated muscle with smooth muscle. So they allow you to connect to those proteins that allow for control of the muscles. Okay, so how does the cell know? How does the cell know that it's in a particular tissue and that it needs to splice an exon out or keep an exon in? There are regulators that are produced. And these generally these are proteins connected with RNAs, again, okay? So there are splicing regulators that are only produced in certain cells under certain conditions. Some of them are regulators that basically say splice out this exon. And some of them are regulators that say keep that exon in. Okay? We actually have genetic diseases that come from problems with those splicing regulators. So there's a particular disease that's called spinal, it's a myotrophy. Myotrophy basically means your muscles are not as good. And what it comes from is your inability to produce the adult form of the proteins that you need in striated muscles. And so you're stuck with the childhood form, the form that is not necessarily good for an adult body because it's really adapted for the small body that you have as a baby. And so they never get to the adult proteins because their splicing factors are not correct. So realize we actually have diseases that are basically regulated by splicing factors. So the splicing factors that I want you guys to know are HNRNPs and SRs. Okay? They are found in some form in every cell. But there's a whole bunch of them. So there's a big family of proteins, a whole bunch of them that are specific for different cells and different genes. They work with the spliceosome. Here's your spliceosome proteins, the U1 and the U2. They will work with the spliceosome to basically dictate whether or not an exon is skipped or whether it's included. So the HNRNP promotes exon skipping. So if you've got those on your exon, basically what will happen, it will inhibit the spliceosome in that area so that you have to basically go from here over to there and skip that exon. The SR proteins will basically enhance the binding of the spliceosome next to that exon. And so they'll encourage you to keep in that exon. So if you have that one, you'll go from here to here, include this exon, and then go from there to there. And remember that these are added. When are splicing factors added to the RNA? During transcription. When does splicing occur?
Or it leaves the nucleus.
Next to the ribosome? No.
Before it leaves the nucleus.
Sorry?
Before it leaves the nucleus.
Before it needs the nucleus. Okay, splicing has to occur before it leaves the nucleus. So that small period of time after transcription, but before it leaves the nucleus is when splicing actually occurs. Probably actually during the time of transcription, because they're added during transcription, and as you're going down, these are long RNAs. As you're going down this RNA, it's probably already starting to splice down here. Because why wait?
So it's like transcription.
Okay. So there's a number of diseases that basically have splicing mutations that involve splicing mutations. One of them is what's called a cryptic splice site, which basically there's a mutation in the gene that makes it look like there's a splice site that's abnormal, a splice site that's basically in the middle of an exon. If that occurs, you'll splice it in properly. So you'll cleave off a little bit of that exon, and you may actually change the reading frame of the protein, which means that your protein may be nonsense at the end. Okay. We'll come back to this proposed treatment because I've got a few things to talk about here. But I want to talk about a specific disease that involves a splicing mutation. So this disease basically is muscular dystrophy. X-linked disease. So it's on the X chromosome. Males only inherit one copy of this gene. Females inherit two copies of the gene. If the one copy that males inherit is bad, they will have a form of muscular dystrophy. There are two different versions of this type of muscular dystrophy. One is called Dystonia, and the other one is called Becker. I assume they're named after the people that kind of wrote about the disease first. I don't know that for sure, but I think that's probably true. Okay, now, the protein that's involved here is called dystrophin. And this is it. It basically anchors between your actin filaments and the membrane of the cell. It's a really, really, really big protein. It's probably the biggest protein that we have. I think that the size of the gene is like 1 million plus bases. It's a freaking big gene. Write that on a test. Okay? It's a really big gene. So it's got all of these subunits. You can see all of these different exons. So all of these blue boxes are exons. Exon, 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 exon. And then that big box at the end is the part that links here to the membrane. And then the stop is the stop codon. Remember that RNAs basically go beyond the stop codon and start before the start codon, right? Because you've got untranslated regions on both sides. Okay. So that basically, those are all the exons. And then you get to the stop codon. Now it turns out that in Becker's muscular dystrophy, there's a large deletion of all of those exons. But what happens is you make a smaller protein, but it still can connect from the nucleus to the actin filament. Okay? So what this protein allows you to do is to basically detect tension in the muscles and basically respond to that tension through the extracellular matrix and the membrane. So if you've got a smaller protein, your muscles are constantly much more tense and they start to degrade. But it's still a fairly mild disease. You might end up with crutches, or you might end up in a wheelchair, but progressively that will happen in the course of a fairly long life. Okay? If you have Duchenne muscular dystrophy, most people will die in their 20s. And you'll progressively get weaker and weaker and weaker in your teenage years. So it's a much worse disease, but if you look at it, it looks like a much smaller deletion. But what happens here is when you go from that exon to that exon, the reading frame is disrupted and you get a really premature stop. And so what it does is it changes the splice site. So rather than basically going from here to here, you basically go from there to the middle of an intron and you connect to that as if it was an exon. Your cell doesn't know any better. You get a premature stop codon, and basically what you have then is this long protein that cannot connect to the cell membrane. So it's kind of flapping in the breeze in the middle of your cell, the big long protein. Gotta go and help. Oh. And because it can't basically allow for your muscles to detect tension, it's going to break down much more rapidly. So sometimes a smaller deletion actually can lead to worse consequences. So in this case, basically both of these are going to disrupt splicing a little bit. That one's going to disrupt it in a worse way because it leads to a premature stop. Okay, so if we wanted to treat this disease, one way to start to treat it might be to basically have an oligonucleotide. An oligonucleotide is a small kind of nucleotide. Oligo means small. They go in the opposite direction to the RNA. And what they would do is it will cover up this particular splice site right there, hopefully allowing you to go from there to the end with skipping that exon. But a small oligonucleotide going in the opposite direction might be a proposed treatment for this. You might also think about introducing this gene, a good copy of this gene, into the muscle cells, but it's a freaking big gene, as I told you. So it's hard to introduce that gene. So there's a search on for treatments for this disease because it's a relatively common form of muscular dystrophy. And it's so catastrophic for the people that suffer from this. So if you were the parent of a child with this, you would probably know really early on that your child suffered from this disease. And you probably would realize that your child only has a small amount of time to live. And there are not really effective treatments as of yet for this particular disease. Okay. All right. There are a couple of other types of post-transcriptional regulation. And one of them basically is to have things bound either the 5' untranslated region or the 3' untranslated region. Okay. So there are things that can be stabilized because you've got proteins bound to that, or they can be destabilized. So you can stabilize your RNA or destabilize your RNA by having specific proteins that bind to the RNA. In that case, you'd always be making the RNA in the cell, but what would happen to your protein levels? I stopped paying attention to this a long time ago. Always make the RNA. What happens to your protein levels? Go up. Your RNA goes down. Oh, sorry. You weren't paying attention, were you? All right, now I know the truth. Ah, no one's listening. All right. So if you destabilize your RNA, their protein levels go down. If you stabilize your RNA, the protein levels go up, right? That makes sense. Okay. Your gene may be, the transcription may be the same, but your RNA levels will change. Okay. What we're going to talk about. Anya will go through this a little bit more. The last thing to talk about is going to be RNA silencing or RNA interference. I remember when this was discovered. This was discovered about 2000, the year 2000 or so, which doesn't sound like a long time. It does sound like a long time ago. How many of you guys were born in the year 2000? We're alive in 2000? Okay, one. That's it. That's very sad.
No, you're
Oh,
saying.
man. Okay. RNA interference is a very cool process. I won $500 off this. I bet. Working in the lab, I said I could make this work. I made it work. Got $500. It was a lot when I was a graduate student. Okay, so RNAi. So inside your cells, you make RNA.
Yeah, but it's I found it today. It was $400.
Remember that your RNA has a start codon and a stop codon. And these two untranslated regions, 5' and 3'.
I won't want it.
Okay. And then what's on this side? That's your 5' side. What's on this side? Your RNA. Yeah. Okay. What's on this side? Poly A tail. Okay. Now, we thought you made RNAs. RNAs got spliced. They went up, got translated. Everything was fine. Turns out, your cell makes a heck of a lot of things that are called RNAis, or siRNAs is another name for them. Okay, or miRNAs is another name for them. So microRNAs, siRNAs, or RNAi. Okay. Our cells naturally make this. What they are basically are small genes that have their own promoter. They have their own promoter, the plus one side. And basically you just make them into RNA. You never translate them into proteins. When you make them into RNA, they double back on themselves and they make this hairpin loop. And there's about 20 bases that are connected together in that hairpin loop. Okay?
That's not a lot.
These are basically these small RNAs, never translated into protein. There is then an enzyme that's called dicer. It's a good name for an enzyme because that's exactly what it does. It basically turns these into this small little piece of RNA. That small little piece of RNA is very specific for one of these two regions, the 5' or the 3' untranslated region. or maybe potentially part of the rest of the RNA. Each of these will have a subset of RNAs that they regulate. Not just one, but a lot of them that will have the same sequences here or here. Okay? What they do is they bind to a protein that then allows them to find the complementary sequence to one side of this double-stranded piece of RNA. Okay? That little enzyme that does this is called the RISC complex. RISC. What it allows basically is for RNases to then come in and degrade this RNA. Or if the RNases do not degrade the RNA, it cannot be translated. So either basically it's not translated, or it's completely degraded. Okay. That's another way to control gene expression, because now you can transcribe it all you want, but are you going to make a lot of protein? No, thank you for listening. Okay, we're not going to make a lot of protein. Okay, I know 5 minutes left, maybe 4. So we're not going to make a lot of protein here. Okay, so this siRNA, this microRNA, is a good way to basically get rid of your RNAs and not produce protein from them. Another layer of complexity. Now, what's really cool about this is I, as a scientific researcher, can use this in the lab and get rid of the RNAs I don't want. Okay? And even maybe I, as a medicine person, a doctor, what are those called? Liker in Ukrainian. I, as a doctor, can basically inject these into my patients, and maybe they can be cured of their disease if the RNA is the problem. Okay, I'm gonna actually end this with the story of Henry. Let's talk about Henry. He's one of these. On CNN the other day. You can actually pull it up on CNN. There we go. Okay, so meet Henry. Isn't he cute? I know. I gave him money because
Henry. Guys.
you only give money to cute ones. I hate to say it. So anyway, so this cute little cow dollar is going to die. I'm sorry to say.
Whoa. Whoa.
Yeah, it's good money. Yeah. Did you see it? Okay, yeah, good. He's trying to raise money. So this cute little toddler is most likely going to die. He constantly has seizures. He's like 3 years old now. He has been constantly having seizures all of his life. There is no treatment for this disease. Okay. The family believes that they have the possibility of a treatment, but they need about $3 million because it's a very rare, it's a treatment that's never been used before. It needs to be specifically designed for him. It needs to be tested. It needs to be then put into his brain. And they think that that will treat his disease. What they're working off of is what we talked about before, using basically an antisense RNA that would work kind of like this guy, would work kind of like a microRNA, but it would be an RNA that's going this way to bind specifically to his defective RNA. Causing that defective RNA then to go down and hopefully treat his disease. I don't know how they're going to plan to administer it, maybe by a virus, maybe by some other means. But they need like $3 million to even try this. They believe if they can get it to work, they may save their toddler. Keep baby lives. And it may help to then provide a proof of concept to treat other people suffering from other diseases. That's what they're looking for. There you go. Experience, as far as I know, I think
It was a complete failure. We're never looking into this again.
they have raised $580,000 Plus another $500,000 that they got from someplace else. They have 2 million to go. So there you go. They're a little way there.
She's gonna get up there.
You never know until you start.